Imagine you are terrified.
Right.
You're walking down a completely dark street, you hear this sudden noise right behind you, and instantly your palms start to sweat, and your heart just feels like it's going to hammer its way right out of your chest.
Oh, yeah, we've all been there.
Yeah, and we have always been told that sequence of events is strictly top-down. Like, your brain senses the danger, it screams orders down a nerve cable, and your heart, which we essentially treat as this dumb, mechanical pump.
Right, just a machine.
Exactly, just blindly follows the instructions to beat faster.
Yeah. And I mean, that is the traditional textbook view, absolutely. The heart is treated as a totally passive receiver of commands.
But the biological reality we're actually uncovering right now completely upends that entire hierarchy.
It really does. It's wild.
Which brings us to the core of today's deep dive. We are exploring a completely groundbreaking 2026 paper published in the journal Cell by Zhu and their colleagues.
Such a fantastic paper.
It really is. They have mapped out something called the intrinsic cardiac nervous system, or the ICNS. And to put it simply, your heart literally has its own little brain.
Yeah, a literal brain in your chest.
It is not just taking orders from the head. It's this local processing center making these active life or death decisions on the fly.
It is a massive paradigm shift, honestly.
Yeah.
This paper is such a milestone because it doesn't just theorize that this little brain is down there.
Right.
The researchers actually went in and decoded its entire molecular architecture. Like they mapped out exactly how it functions actively to keep us alive, second by second.
So I want you, the listener, to keep this in mind the next time you feel your heart race or maybe flutter or skip a beat. That might not be a top-down command from your head.
Probably isn't.
Right. That might be your heart's own local brain evaluating a stressor and deploying its own countermeasures without ever even consulting you.
It's doing its own math.
Yeah. So our mission today is to decode this cardiac brain, map out its two primary neuron types, and uncover the astonishing roles they play in our daily survival and stress responses. Okay, let's unpack this.
Well, to really grasp how this little brain operates, we first have to figure out what it's actually made of, you know?
Sure.
Because historically, the ICNS was just a total nightmare for scientists to study.
Really? Why was it so hard?
Oh, it looks like this incredibly sparse, tangled web of microscopic nerves, and it's just plastered all across the surface of the heart muscle, all mixed in with fat and connective tissue.
So it's basically a messy haystack. How did Yus and their team finally untangle it?
Well, they used a highly advanced technique called single-cell RNA sequencing.
Oh, okay.
Yeah, they took heart tissue from mice, dissolved all that connective tissue, and managed to isolate exactly 2,870 individual intrinsic cardiac neurons.
Okay, wait. I want to make sure I understand the RNA sequencing part of this.
Yeah, go ahead.
Is that essentially taking each of those 2,870 cells, ripping them open, and like reading the specific genetic instruction manual inside to see what that particular cell does for a living?
It's actually a brilliant way to describe it.
Yeah.
Yeah, exactly.
Awesome.
By reading the RNA, which is the active copy of the DNA being used at that very moment, they could see exactly which genes were turned on or off in every single isolated cell.
So they get a profile of what each cell is built to do. Now, a lot of people try to compare this cardiac brain to the gut's famous second brain, right? The enteric nervous system.
Right, people love doing that. But the genetic sequencing here revealed a totally different architecture.
How so? I mean, I imagine a local brain needs to be pretty complex to run an organ.
Well, see, the gut's nervous system is incredibly complex. It is packed with sensory neurons and all these diverse interneurons that chatter back and forth all day long.
Because it's breaking down food, absorbing things, moving stuff along.
Exactly. But the heart's little brain, by contrast, is aggressively streamlined. Like it completely lacks those classic sensory markers.
Oh, really?
Yeah, and it doesn't even have its own adrenergic adrenaline-producing neurons.
Wait, so if the gut's brain is this massive, sprawling internet network with like billions of varied connections, the heart's brain is more like a highly specialized dual-core processor.
Yes.
Like stripped down, no fluff, just designed to execute a critical job without any lag.
That dual-core analogy is spot on because the sequencing revealed exactly two major genetically distinct subtypes of these neurons.
Okay, what are the two cores?
So the first subtype is defined by a gene called Nb, which produces neuropeptide Y.
NPY.
Got it. Right. So we will call them N-positive neurons. The second subtype is marked by Ddah1, which is an enzyme involved in nitric oxide signaling.
DAH1. Okay, so we will call those Ddah1 positive neurons.
Well, exactly.
So I'm picturing this messy web of nerves on the heart. If this is a single integrated local brain, I assume these two types of neurons just like overlap everywhere, just sending signals to the exact same parts of the heart muscle.
What's fascinating here is that the physical map looks completely different than you might expect.
Oh.
Yeah, to see where these wires actually go, the researchers use these hollowed out, completely harmless AAV viruses.
Like virus delivery trucks.
Basically, they loaded these viral delivery vehicles with a fluorescent tracking dye and injected them so that only these specific neurons would glow brightly.
But wait, the heart is a thick, opaque muscle. How do you see glowing microscopic nerves inside a solid piece of meat?
So they used a tissue clearing method called iDisco.
iDisco.
That sounds fun.
It's incredible. By soaking the heart in a specific series of chemical solvents, they dissolved all the opaque fats and lipids without damaging the cellular structure at all.
Whoa!
Yeah, the entire heart becomes completely transparent, like a glass sculpture.
That is wild.
Right. So under a 3D microscope, you can literally trace the glowing nerves through the clear tissue, and it revealed a total strict division of labor between the two cores.
A transparent glowing heart map. Where do the glowing wires actually go? Well, the NP-positive neurons cast an incredibly wide net.
Okay.
They send these sweeping projections down to the SA node, which is the heart's natural pacemaker,
Right.
the AV node, the ventricles, and they form this very dense, tight web encircling the aortic root.
And the aortic root is the massive base of the artery pumping blood out to the whole body, right?
Exactly.
Got it. So the NP-positive side of our dual-core processor is like the wide-angle lens. It touches almost all the major plumbing and electrical hubs.
Yes.
So where does the Ddah1 positive side go?
They are drastically restricted, like totally different. Their projections bypass the main pacemakers almost entirely.
No kidding.
Yeah, instead, they strictly target very specific anatomical hubs, primarily the pulmonary artery and the ostia, which are the openings of the pulmonary veins.
Okay, let me make sure I'm wrapping my head around this before we move on. Let me just give a quick bulleted summary for you listening.
Go for it.
So we have established that the heart has a streamlined, dual-core local brain, two distinct neuron types. Number one, the Npy-positive neurons cast a massive net across the heart's main plumbing and pacemakers.
Right.
Number two, the Ddah1 positive neurons stubbornly sit only at specific vascular openings, like the pulmonary vein.
Spot on.
Which leads me to a question for you, the listener. Think about this. If you were designing an engine, why on earth would you build two entirely separate control systems? One that touches everything and one that is hyper-focused on just one or two valves. Keep that question in mind as we keep going.
It's a great question. And to answer that, we really have to look at what happens when these systems are engaged in the living animal.
Let's do it. Let's look at that broad network first, the NP-positive neurons.
So the researchers discovered that these neurons receive their incoming commands from the vagus nerve.
The vagus nerve, okay, that's the body's main parasympathetic highway, right? The rest and digest pathway.
Yes, exactly.
So these NP neurons act as the brakes to slow things down.
The data strongly supports that. And they actually proved it using optogenetics.
Using light.
Yeah, light. They genetically modified the NP neurons in mice so that they would only activate in response to blue light.
Wow.
So when they shone a blue laser directly onto the heart, the NP neurons fired and the heart rate instantly dropped.
Just like hitting a brake pedal.
No.
But they also proved it in awake, moving mice too, right?
They did.
Because the paper mentions chemogenetics, which sounds entirely like science fiction to me. Are they somehow feeding these specific microscopic nerves like drugs while the mouse is running around on a wheel?
Essentially, yes, they are. Chemogenetics relies on a custom lock and key system.
Okay.
They engineered the NP neurons to display a synthetic receptor on their surface. Think of it as a custom-built keyhole called a dread.
Dread. So it's a keyhole that isn't normally there.
Exactly. And this keyhole is entirely inert. It ignores every single natural hormone or chemical in the mouse's body. Adrenaline, dopamine, it doesn't care.
It's just sitting there waiting.
Right. It only unlocks when the scientists inject a highly specific synthetic drug.
So they can inject this drug into a perfectly normal acting mouse. The drug circulates, hits only that specific custom keyhole on the NPy neurons, turns them on, and the heart rate immediately slows down.
Yep. Instantly.
That is incredibly elegant.
It is a beautiful demonstration of baseline calming control. But The researchers needed to know how crucial these breaks really were to daily survival.
Like what happens if they break?
Right. So they devised a highly targeted ablation experiment. They genetically engineered the mice so that only the Npy neurons had a vulnerability to diphtheria toxin.
So they basically put a self-destruct button on just those broad network neurons.
Yes. They injected the toxin,
Right.
and it cleanly, invisibly wiped out the entire Npy network, leaving all the rest of the heart completely untouched.
Here's where it gets really interesting. You take away the brakes. Logically, the heart should just beat a little too fast, right?
Well, that's what you'd think.
Maybe the mouse gets a high resting heart rate, but the paper says this experiment was fatal. The mice died within a week.
Within a week, yeah.
And it wasn't a sudden electrical short circuit or something. Their heart rate and blood pressure just progressively collapsed over several days.
It was a massive puzzle for the research team. I mean, why does losing a nerve that slows the heart result in fatal cardiac failure?
It seems completely backward.
Totally backward. To solve it, they had to isolate the heart muscle entirely from the rest of the body using an ex vivo Langendorf setup.
Wait, ex vivo? Like outside the living body, they took a failing heart out of a dying mouse and hooked it up to a machine?
Yes.
How does it keep beating?
Well, the Langendorf rig is a classic cardiovascular research tool. You take the heart out of the chest cavity, hang it on this delicate rig, and you basically pump an oxygenated, nutrient-rich fluid backward into the aorta.
Backward.
Yeah, this artificial pressure forces the fluid down into the coronary arteries, feeding the heart muscle perfectly, entirely independent of the body's own blood pressure.
Oh, I see. So the heart is suspended there, just beating away on its own.
Exactly.
What did they see when they put these Npy-ablated failing hearts onto the Langendorf rig?
The heart's pumped flawlessly.
Flawlessly.
Yeah, the muscle contractions, the ventricular pressure, the pacing, everything was totally normal.
The heart muscle itself wasn't broken at all. Outside the body, on artificial life support, it was a perfectly healthy pump. So why on earth were they dying inside the living animal?
The answer lies in that dense web of nerves we mentioned earlier, the one encircling the aortic root.
Ah, the plumbing at the top.
Right, because inside a living body, the heart faces a really unique physics problem. It cannot feed itself while it is actively squeezing.
Wait, really?
Yeah. During systole, when the heart forcefully contracts to push blood out, the pressure actually squeezes the coronary arteries shut.
Wow, I never thought of that.
So the heart only receives blood during diastole, when it's relaxed.
I'm trying to visualize the plumbing here. When the heart pumps, it blows the aortic valve open, shoving all this blood out to the body. Then it relaxes, and the valve snaps shut.
And when that valve snaps shut, the blood bouncing backward against it causes the thick elastic walls of the aortic root to bulge outward and then violently snap back to their original shape.
Like a rubber band.
Exactly. And that elastic recoil maintains enough ambient blood pressure to shove blood sideways into the coronary arteries. That mechanical pressure snap is called the dicrotic notch.
Ah, I see. So it's like a thick, pressurized rubber bulb pushing water into small side pipes.
Right.
When you squeeze and release the thick rubber bulb, it violently snaps back to its original shape, and that force is what pushes water down those side pipes.
Precisely. And the researchers discovered that the Npy-positive neurons are actively, constantly providing an electrical tone to the aortic root.
To maintain that rigid rubber bulb elasticity.
Yes. When the toxin killed the Npy neurons, the aortic root completely lost its tone.
It lost its snap. The thick rubber bulb basically turned into a flimsy plastic grocery bag.
That's a great way to put it.
So when the blood bounced against the closed valve, the flimsy bag just ballooned outward. The dicrotic notch vanished, the pressure plummeted, and the side pipes, the coronary arteries, ran completely dry.
Yep. The heart muscle starved itself of blood simply because the plumbing lost its mechanical tension.
It is a profound revelation. It proves that the Npy-positive neurons are not just sending a simple slow-down electrical signal.
Far from it.
They are actively, continuously managing the physical, mechanical plumbing of the cardiovascular system just to sustain baseline life support.
It's a completely new way of looking at it.
So let's take a beat to summarize this piece of the puzzle real quick.
Go for it.
Number 1, we know the Npy-positive neurons slow the heart rate. Number 2, we now know they actively control the mechanical pressure of the aortic root. And number 3, without them, the plumbing structurally fails and the heart literally starves itself.
Absolutely brutal.
So I ask you, the listener, does it surprise you that removing a calming rest and digest nerve causes a fatal mechanical failure rather than just a fast pulse? It really forces us to entirely rethink what baseline health actually means in the heart.
It absolutely rewrites the textbook. But, that only explains one half of our dual-core processor.
Right, the wide-angle lens.
Yeah. So if the NB positive network is this broad caretaker keeping our plumbing pressurized at baseline, what on earth is the highly restricted Ddah1 positive network doing, sitting almost exclusively by the pulmonary veins?
Right, because they aren't touching the pacemakers, they aren't touching the aorta. What is their job?
Well, to figure that out, the researchers first looked at where Ddah1 gets its orders.
Okay.
Unlike the Npy network, which receives calming vagal input, the Ddah1 network receives direct sympathetic input.
Oh, the fight-or-flight pathway.
Exactly. These are the raw, unfiltered fight-or-flight panic signals. And they map this out using a highly specialized rabies virus tracing technique.
Rabies. That sounds incredibly dangerous to just inject into a heart.
Oh, it is a safe, heavily modified version of the virus. But rabies has this unique, frankly terrifying natural ability. It travels backward, up the nervous system.
It's like against traffic.
Yeah. If you get bitten on the hand by a rabid animal, the virus jumps from nerve to nerve, traveling up your arm, into your spinal cord, and eventually into your brain.
Creepy.
Very. So the scientists modified the virus so it would only glow green, and they inserted it strictly into the Ddah1 neurons in the heart.
So because it travels backwards up the neural pathways, it acts like a glowing breadcrumb trail, showing exactly which wires in the body are plugging into the Ddah1 neurons.
Precisely.
Where did the glowing trail lead?
It led straight back to the stellate ganglion, which is this massive nerve hub in the chest responsible for pumping out sympathetic, adrenaline-driven stress signals.
Wow. So the Ddah1 neurons are explicitly hardwired to receive panic signals.
Yep.
Okay, so they were wired for stress. What happens if you kill them off? Do the mice go into immediate cardiac collapse like they did with the Npy ablation?
Well, the researchers used that same diphtheria toxin trick to wipe out just the Ddah1 network.
Yeah.
And the result was absolutely nothing.
Wait, nothing?
The mice were completely fine.
Yeah.
They just sat in their cages, their resting heart rates were normal, their plumbing worked perfectly. At baseline, the Ddah1 neurons seemed to do nothing at all.
But they are wired for the fight-or-flight system. You wouldn't notice a broken fire alarm until there is a fire.
Exactly. They had to stress test the mice.
How do you stress test a mouse?
And the stress tests were rigorous. They started with psychological stress. They took the mice and simply restrained them inside a clear plastic tube for 20 minutes a day.
Okay, not pleasant, but doesn't sound lethal.
Right, a normal mouse hates being in a tube, but it survives the experience perfectly fine. But the Ddah1 ablated mice, the vast majority of them died within days.
Just from the tube.
And they didn't die later in their cages. They died suddenly during the 20-minute restraint period.
They basically died from being psychologically stressed. What was happening inside their chests?
So the researchers built a specialized, miniaturized ECG chamber to monitor the mice's heart rhythms in real time while they were in the tube.
Okay, what did the readouts show?
The readouts were chaotic. Under stress, without the Ddah1 neurons, the electrical grid of the heart went into total meltdown.
Jeez.
They developed malignant arrhythmias. They suffered from severe AV block, which is where the top chambers of the heart completely stopped communicating with the bottom chambers. And then sudden cardiac arrest.
And I remember reading, they didn't just use psychological stress, right? They used intense physical and chemical stress, too.
They did, yeah. They subjected the mice to chronic heat stress, keeping their environment at 37 degrees Celsius, which is about 98 degrees Fahrenheit, for a whole week.
Wow. Normal mice can handle that.
Normal mice can regulate their core temperature and handle it just fine.
Right.
But the Ddah1 ablated mice died rapidly of sudden cardiac events.
Wow.
Finally, they simulated an extreme catastrophic panic attack. They injected the mice with a massive near-lethal cocktail of epinephrine, because adrenaline, and caffeine.
A massive adrenaline dump. A normal mouse's heart must have been beating at lightning speed from that.
It was. But a normal mouse's heart can usually stabilize its rhythm and survive the surge. When they gave that exact same chemical cocktail to the Ddah1 ablated mice, their hearts instantly went into total electrical dissociation and they died within minutes.
That is brutal. So let me make sure the mechanism is crystal clear here. Sympathetic stress. whether it is psychological panic, physical heat, or a chemical adrenaline dump, naturally kicks the heart's electrical system into high gear.
Right.
If that high gear pushes too far, the entire electrical grid essentially shorts out, causing a fatal arrhythmia.
Yes.
It sounds like the Ddah1 positive neurons act as a local surge protector.
That is a perfect analogy. They are a biological circuit breaker built directly into the tissue.
So when the stress spikes, the Ddah1 surge protector clamps down on the local electrical grid to stop it from catching fire. And the researchers proved this with a rescue experiment, didn't they?
Yes, using chemogenetics again.
The synthetic lock and key.
Exactly. Instead of deleting the neurons, they engineered the Ddah1 network with that custom chemical lock. They injected the custom drug to constantly stimulate the neurons, essentially locking the surge protector in the maximum power position.
Okay, so the surge protector is fully engaged.
Right. And when they subjected those mice to the lethal adrenaline and caffeine cocktail, their survival rates skyrocketed. Activating the local circuit breaker saved them from fatal arrhythmias.
If we connect this to the bigger picture, remind me where these Ddah1 neurons are located physically again.
Primarily clustered around the ostia, or the openings of the pulmonary veins.
Right, and from a clinical cardiology standpoint, the pulmonary vein is incredibly notorious.
Oh, it is one of the most problematic areas of the human heart. It is globally recognized by cardiologists as a massive hotspot for triggering dangerous arrhythmias, particularly atrial fibrillation.
So by discovering that the Ddah1 neurons sit exactly at this known electrical hotspot and proving that their sole job is to buffer against sympathetic overdrive, we suddenly have a molecular explanation for why that specific piece of cardiac real estate is so vulnerable when we are stressed.
Exactly. It completely demystifies the chaotic landscape of stress-induced arrhythmias.
Okay, let's track the logic on this third section for a quick recap.
Sure.
Number one, we know Ddah1 positive neurons receive fight-or-flight stress signals. Number two, we know they sit precisely at known arrhythmia hotspots. And number three, we know that without them, routine stress causes fatal electrical short circuits.
Perfect summary.
So I want you to think about the last time you were terrified or exercising really heavily or maybe incredibly stressed out about a work deadline.
We all have those days.
We do. And how hard was your local Ddah1 surge protector working in the background to keep your heart rhythm stable while adrenaline just flooded your system? It is awe-inspiring.
It really moves us far beyond the idea of a dumb pump. We are looking at a highly sophisticated, integrated survival system.
So what does this all mean? If we synthesize everything from this deep dive, we're looking at a beautifully streamlined dual system brain inside your chest.
Yes.
On one side, you have the Npy positive neurons. They're your baseline caretakers. They cast a wide net, monitoring your plumbing, maintaining the mechanical tension of your aortic root, and ensuring that with every single beat, the diastolic pressure snaps back to physically push blood into the heart muscle.
Beautifully said. And sitting right alongside them, you have the Ddah1 positive neurons. These are your emergency responders.
The surge protectors.
Exactly. They are totally quiet at rest. But the split second you enter a fight or flight state, they activate. They physically guard the heart's most vulnerable electrical hotspots, buffering against the adrenaline surge to prevent a lethal short circuit.
It is an incredibly elegant division of labor. But practically speaking, how does decoding this dual-core processor actually change how we treat human heart disease?
Well, currently, our treatments for severe arrhythmias can be quite blunt.
Blunt? Like how?
If a patient has debilitating atrial fibrillation, a really common procedure is catheter ablation.
Okay.
A surgeon threads a wire into the heart and literally burns away nerve tissues around the pulmonary veins to stop the erratic electrical signals.
They just burn the tissue.
Yeah. It is effective for some, but it is highly nonspecific. We are burning everything. The bad tissue, sure, but also potentially the baseline caretakers and the emergency surge protectors.
Wait, so we might literally be burning away the Ddah1 circuit breakers by accident, leaving the heart more vulnerable to future stress?
Precisely. We just didn't know they were there before. This research paves the way for the future of precision cardiology.
Wow.
Instead of blindly burning tissue, imagine using targeted gene therapies or highly specific pharmacology. If a patient has perfusion issues, we could selectively boost the Npy plumbing regulators. If a patient is prone to panic-induced arrhythmias, we could design drugs that specifically stimulate the Ddah1 surge protectors. We are basically moving from using a sledgehammer to a microscopic scalpel.
This raises an important question, though.
Oh, what's that?
Well, if the heart has its own dedicated local brain, one that actively evaluates stress signals, senses its local environment, and makes immediate life or death decisions.
Right.
What happens during a heart transplant?
Oh, wow. Yeah, that is the million-dollar question.
Because I hadn't thought of that until just now.
It's fascinating because when a surgeon removes a donor heart, they completely sever the vagus nerve and the sympathetic cables connecting it to the original brain.
They snip the wires.
They snip them all. They drop that heart into a new chest, stitch up the plumbing, and shock it back to life. But those neural cables back to the brain are never reconnected.
Never.
Never. That transplanted heart is physically and neurologically isolated. So how does this local intrinsic brain survive, adapt, or rewire itself to protect its new host from stress without any top-down input?
That is just, it makes you look at every single transplant in a completely new, mysterious light.
It really does. It's a whole new frontier.
That is incredible to think about. A little isolated brain suddenly waking up in a completely new environment, having to manage the plumbing and the surge protectors all on its own.
It's a tough job.
It is. So the next time you feel your heart leap into your throat before a big presentation or flutter when you are startled, don't just write it off as a mechanical pump struggling to keep up with your brain.
Give it some credit.
Yeah. Take a second to appreciate the hidden local intelligence working furiously in the background, making complex life-saving calculations in real time just to keep you standing.